Hello, I'm Leena Malayil, a postdoctoral fellow at Dr. Amy Subcota's lab at the University of Maryland School of Public Health. I'm extremely delighted to present my talk titled Source Tracking Metabolically Active Bacterial Communities from Rainwater to Produce. Rooftop harvested rainwater is gaining increasing interest as a potential irrigation water source. Previous studies have indicated the presence of both biotic and abiotic factors affecting rooftop harvested rainwater quality. However, very few studies have investigated the potential transfer of microorganisms, particularly metabolically active bacteria, from rainwater to irrigated soil and produce. To bridge this knowledge gap, we collected 364 samples from a vegetable rain garden in Maryland, USA from June to August in 2018 and 2019. Among the samples collected were water from the first flush tank, secondary tank, municipal water and ambient rain. Soil and produce were also collected from the 5 soil beds as represented in this figure. The subsamples were treated with a DNA label 5-bromo-2-deoxyuridine, in short BRDU, which helps to distinguish between live and dead bacterial communities present in a particular sample. DNA from treated and non-treated samples were then extracted and PCR amplified for the V3-V4 hypervariable region of the 16S rRNA gene and sequenced using the Illumina HiSeq. Sequencing data were then analyzed using CHIME, multiple R packages, and Source Tracker. Here, I give you a glimpse of the results. We first looked at the shared and unique bacterial profiles between the different sample types among BRDU-treated samples. We observed Pseudomonas, Sphingommonas, Aeromonas, Arthrobacter, and Bacillus as the predominant genera in these sample types. Then we looked at the average relative abundance of the top 25 bacterial communities in the different waters that were collected. That is the ambient rain, the first flush tank, municipal water, and the secondary tank. The x-axis over here represents the average relative abundance, while the y-axis represents the taxonomy. The red bars here indicate BRDU-treated samples, while the green is the non-treated. It is very much evident over here that the first flush tank and the secondary tanks had more metabolically active bacterial communities. Next, our interest was to find out if there was a potential transfer of microbes from the irrigated water to produce. Here, the pie charts represent the likely source of microbial communities on the surface of produce taken from each of these soil beds. Beds 1 through 3 shows us that the first flush tank to be the major contributor. To sum up my study, we found diverse metabolically active bacterial communities shared between the different sample types. And next, we found that the first flush tanks and the secondary tanks were the major contributors to the bacterial community of the irrigated produce. I would like to thank Conserve, a Center for Excellence at the Nexus of Sustainable Water Reuse, Food and Health, funded by USDA NIFA. Lastly, but not the least, I would like to thank the National Council for Science and the Environment for giving me an opportunity to present this talk. I hope you all liked the talk, and if you have any questions, please feel free to email me lmalayil@umd.edu. Thank you.